Hello everyone, my name's Amy and I'll be the person taking you through this tutor session today. Most of you kind of know me or should have heard from me. Just to tell you a little bit about myself, this is me. I do live in Salt Lake City. I have a bachelor's degree in English and a master's degree in education and policy. And I tell you this because I actually did not graduate from high school. I flunked out of high school. So I've always been a non-traditional student and always one step behind until I started getting ahead. And I tell you this to let you know that you can get past these, and I'm going to help you do that. And you can do anything that you set your mind to. So before I do get started, I do want you to grab a pen and paper if you don't already have one. I'm going to ask you to do notes along the way. I'm going to give you important keywords and important concepts that you'll want to write down. I'll also have a study guide that's going to complement this video afterwards, so you will have a reference sheet. And also, before we get in, the slides are going to seem to jump around a lot because this is the Science OGT. And with the Science OGT, there are several different disciplines of science that you need to know a little bit about. You have to be a master of none, but you have to know a little bit about each of these disciplines. So we'll be doing astrophysics, earth and physical sciences, biology, technology, and all different types of science. So it's going to seem to jump around a lot, but I have studied the OGTs pretty intensely over the last couple of months. I have a super good idea what's on there. And so this slide should really give you a really good idea of what you need to know. So let's get started right away. I'm going to start here with these two different cell types. We've got a eukaryote cell and a prokaryote cell. And you can kind of see right away what the difference is between the two. I'm going to tell you a little bit about this word prokaryote. This means before nucleus. And if you look down here, you see this little jumbled mess here. It looks like a colon. And this is a nucleoid. And it's kind of just free floating in here. It is the control center. It is where all of the DNA is. And then actually, anytime you see this prefix nuclei anything, it always means the control center. It always means it's at the center and it's what controls whatever particle you're talking about. So here's our prokaryote. This is one of the oldest cells on the planet that we know of. And eventually, over time, they evolved into these eukaryotes. And so all of the animals and all living things that you can think of and see, the more complex life, are made out of these eukaryote cells. And if you look at these little bits and pieces here, these are called organelles. You can remember what that is by thinking about your own organs in your body. These are just the bits and pieces of the cell that help it function. Then I want you to notice right away the differences here. And the reason why this cell is evolved is because it has this nuclear membrane and a nucleus, and all of the DNA and all of these processes are nestled all nice and neatly here in this nuclear membrane. And this is what helps these eukaryote cells to evolve. Now we've got a bunch of different pieces. But actually, for the sake of the OGT, we're going to take a look at this prokaryote cell because there's certain things that you need to know about it. So here's a picture and a diagram of bacteria. And so you've got this cell wall. It's exactly what it sounds like. It holds all of this inside, all of this special stuff here. This is all your important stuff. Well, it's all ultimately important, but this is what controls and performs all the functions of the cell. And then here's your nucleoid here. And like I said, this is the genetic materials, is where all your DNA is. This is where all the decisions of the cell are ultimately made. And then these guys carry out whatever this DNA tells them to. And this cytoplasm is kind of the medium that these float around in. And it's exactly what it sounds like. It's plasm. It's a gel-like substance. It actually protects these little important pieces inside of the bacteria, but it also helps synthesize a lot of the chemical reactions inside the cell. And then ribosomes here, these synthesize protein. And the way I want you to remember that is through this prefix here, rib, which is meat, which is protein. So if you can remember that, you'll never forget what a ribosome is. So actually, that might be something you want to write down. You will probably see the word ribosome on the OGT test. So if you know what it is, it's super helpful. But for the sake of the OGT, I really want you to pay attention to this right here. And this is the flagellum of a bacteria. And this flagellum actually is what propels this bacteria cell through any medium it's traveling through. And it does it by waving like a flag, flagellum. So it waves back and forth like a flag, and that's how it moves through. And I actually have a little video here I'm going to show you of what that looks like in person. So this right here, this right here is a plant cell, and you can tell by these right here, these are chloroplasts, and only plants have those. But see how this guy is like waving back and forth here. Here's another one here, and they just wave these back and forth, and then it um, moves them through and side to side. Now this one is also important. I want you to pay attention to it because these little hair-like structures right here, this is called cilia. Here's how you spell it right here. You'll probably see that word again on the OGT, but this is another way that these cells propel themselves through any kind of liquid medium. And these hair-like structures wave and push it through, and you can watch it a little bit more here. But now there are two types of human cells that you can think of. There's one that has flagellum, and that's the sperm cell, and that's exactly how the sperm propels itself upward and moves. And then as far as cilia, our lung cells have cilia, and it's what helps facilitate the movement of oxygen through our lungs. So these are the pieces. So I want you to pay attention to this because here is the question or a similar question that's on the OGT that you do need to know. So make sure you're writing this down. The picture below shows some of the structures in a single-celled organism. Here's what we kind of just went over. The presence of which structure provides evidence that this organism is capable of locomotion. Is it the cell wall, the ribosome, flagellum, or the cytoplasm? If you remember correctly, it is the flagellum, and it waves like a flag, and that's what propels it side to side and forwards and backwards. So we're going to jump now to plate tectonics, and we're going to talk about this for a second. I want you to write down this name, Alfred Wegener. Okay, he was the theorist of continental drift. And the theory of continental drift, which he coined as Pangea, states that all of the land masses were joined together at one time. So Wegener looked at a map and he noticed that all of the countries looked like they were fit together like a puzzle piece. Like if you brought them all together, they would squeeze in kind of perfectly nice. And then also he noticed that there were similar fossils through these different continents that are now oceans apart of animals or plants that should have never existed together. To show you what I mean by that, here we've got this Sinognathus. I don't know how to say that. It was here and up here. And South America is a very tropical climate. And Africa right here is very arid and dry. So this species could not have survived here in these two different climate types. But if these were at one time together, then the species could have survived here. So we have all these examples of different kinds of fossils that exist in these places they should never have been. Well, so Wegener was he was a pioneer of his time, but other scientists did not believe him. And the biggest reason why they didn't believe him is he couldn't explain, he didn't have the first word to explain how those land masses drifted apart. And so they kind of made fun of him, and he kind of had this tragic death where he died in Antarctica. Um, and he died not knowing that eventually his theory was actually proven true. And it was proven true through plate tectonics. And here's a picture of plate tectonics. These are large plates that are embedded within the mantle of our Earth. They are constantly moving. They're moving underneath your feet right now. And they move about 1 to 2 inches per year. So this is excruciatingly slow in geological time. So you can imagine in probably another couple million years, the map of our Earth is going to look really different. These are constantly moving. They slide up against each other. They slide past each other, against each other. When they push into each other, that's how you form mountain ranges. When they slide past, you get earthquakes, you get volcanoes, and this is all of our geological action happening. And so I have this complicated diagram, but there's only a couple of things I really want you to remember about this. So pull out your pen and paper. And I want you to notice these circular motions right here, these arrows going around. This is called convection. Now you're going to see convection several times on the OGT, and they're going to talk about it in different capacities. But basically, convection is the movement of heat or thermal energy, and this is how it's moved. If you think of a pot of boiling water, it boils in a very circular motion, right? That is how heat is transferred through liquids. And so underneath this part of the earth right here, this is not quite solid, but it's not quite liquid. But this is where all this convection is happening. And eventually what happens is you have your molten core here, you have lava, and it comes up, and then you get these underwater volcanoes. So these volcanoes, like they come out, you get lava rock, it cools off around, and this new land is being perpetually formed as the lava rock cools off, and then another volcano, and so on and so forth until you get like land. Well, here's a better one because here's a tiny little island starting to come up above the ocean. So if you were to get into an airplane and take a trip around the world and fly over the oceans, what you would see are these little scattered spots of land. Some of them are inhabited, some of them aren't. There's a lot of islands out there that have nothing on them. They're pretty new in geological terms. They're relatively new, so life hasn't inhabited them yet. But new land is constantly being formed underneath on the ocean floor. And so this right here is called a mid-oceanic ridge. It's something you're gonna see on the OGT, so write that down, mid-oceanic ridge. And this is where new land is being formed all the time. And you can kind of see how it has this ripple effect. So if you were a scientist and you were traveling this way, you would be finding these younger and younger rocks because you're traveling towards land, new land. So that's what I want you to know about here. This is an ocean trench. These are very deep parts of the earth, probably the deepest parts of the earth. And right here you have subduction happening, and then we're pushing up a mountain range here. So this is a question that I think is on the OGT. The cross section below shows the orientation of rock layers in a given area. What geological event most likely caused the folding and faulting of these layers? Tectonic activity, glacial advance, magnetic reversal, or seafloor spreading? Well, your keywords are folding and faulting. These are tectonic activities. And you will see the folding and faulting on the OGTs. Folding is literally like the folding up a land. So if you were to look at folding, you would see like these neat, kind of like two bookends pushing earth together. So it looks kind of crunched up and folded. And then the faulting is more of a displacement of the land. So you think of like an earthquake where land is just shifting and moving. That's the folding and faulting. And that's what's happening here. So the answer to this is tectonic activity. And if you haven't seen this on the OGT, if you're taking version 1, the OGT, remember this question, it is on there, and the answer is tectonic activity. So we're going to move on. We're going to stay with geology. We're going to study Nicholas Steno, who is considered the father of geology. And he came up with the law of superposition, which is to say that in a series of undisturbed series of sedimentary rocks, the oldest rocks will be at the bottom and the youngest will be at the top. And to give you a visualization of what that means, you have this guy here and he's digging, 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 and it's like a time machine, right? So he's going back into history. The further down he digs, the older this rock is getting, and the more you can tell about what happened to that section of the earth. So here's a question. In the 1600s, Danish scientist Nicholas Steno studied the relative positions of sedimentary rocks. He determined that sedimentary rocks typically form layer on top of layer, so if undisturbed, the bottom layer of sedimentary rock is the oldest. Today, this idea is known as the law of superposition. This law is one way to estimate the relative ages of sedimentary rocks. The diagram below is a geologic cross-section from Ohio. So one thing I'm always going to tell you about the OGTs is when you see these charts, take a moment to understand them before you move on to the answers, because it's going to, they can't trick you if you understand the graph before you get down there. And part of getting through the OGTs is understanding all the different ways they're going to try to trick you. So let's take a look at this. So the very bottom here, we have granite and then limestone, and we have some sandstone, a little bit more limestone, shale, and then at the very top are these glacial deposits. So according to the law of superposition, the oldest is this granite layer here. And the last geological activity on this slice of land in Ohio are these glacial deposits. So there was a glacier here, and that's the last thing that they can tell that happened here. How could the law of superposition be applied correctly to this cross section? The youngest sedimentary layer was deposited by glaciers. The layers in this cross section are all igneous, so it will not work. The sedimentary layer in this cross section is close to 5,200 years old, and the youngest sedimentary layer can be found at the very bottom of this cross section. So we're going to take a moment to look at these answers, because there's some tricks here that are going to help you if you know these on the OGTs. So you have, first of all, you have an answer here you can eliminate right away. We don't have any kind of reference to time. We have no idea how old this is. No clues given. You can take that out. We don't have enough information for that. Now, igneous rock is actually lava rock. So that diagram we looked at where we have the mid-oceanic ridges, that new rock forming, that is lava rock. Those are igneous rocks. One good way to remember igneous is ignite, which means that these were basically ignited rocks at one time. They were lava that cooled off into these igneous rocks. Igneous rocks actually metamorphosize into different types of rocks over time, but they all start out igneous. That's not true about any of these. These are all different types of rocks. So you have this youngest sedimentary layer was deposited by glaciers, and the youngest sedimentary layer can be found at the very bottom of this cross section. These two answers are completely opposite of each other. So anytime you see two opposites like this, you know that it's going to be one of those twos. They're going to try to trick you. And what I've seen a lot of students do is choose D because they read it too fast. This says the youngest sedimentary layer can be found at the very bottom of the cross section, but they see the youngest sedimentary layer and they select D. But this is the oldest layer. The youngest sedimentary layer is deposited by glaciers. It's at the top. So even if you understand the law of superposition really well, make sure when you do these questions, you're not being tricked by the answers they give you. Slow down, pay attention to the answers, make sure you're getting the right one. Okay, the cross selection below shows a series of rock layers that have been found in all of these land masses: South America, Africa, India, Australia, and Antarctica. Alfred Wegener's analysis of the similarities in these layers led to the conclusion that continental plates float on top of a molten mantle. In undisturbed rock sequences, the oldest fossils will be on the bottom. These 5 land masses were once joined together in a single landmass, or magnetic anomalies are preserved in rocks formed at mid-oceanic spreading centers. So go ahead and answer that in your head. If you answered B, they tricked you. This is Nicholas Steno's law of superposition. But that is not what the question's asking. And this is why I wanted you to write down Alfred Wegener's name. Because if you see his name and or a list of different continents, this is a single landmass question. So they do this a lot on the OGT. They always ask a question about the single landmass, and you can always tell it's a single landmass question because they're going to list off these different land masses here. They're going to show like this. This is a similar geologic history belonging to each of these continents that are oceans apart now. Should never have had the same processes happen. But so that's, and your other clue is Alfred Wegener here. So this is actually see these fine land masses were once joined together in a single landmass. Remember that. They will try to trick you with this. So, because you know this is a true statement. It's just not the statement that's um, supporting it's not answering the question. So if they list this, say his name, you know it's a single landmass question. So we're going to go over this story problem. They usually have two of these on each of the science tests. And you look at them and they might seem overwhelming. They're long, seems like they're complicated. Totally not. And actually, these charts that they give you are usually extremely simplistic. So as long as you understand them, you're going to know how to answer the questions. You just have to take the time to read through them and understand what you're reading. The British peppered moth Biston betillaria occurs in two colorations, light, mixed black and white, and black. Black coloration was first seen in the 18th century moth collections as a rare, highly prized mutant. Black coloration is controlled primarily by a single dominant gene. Before 1850, the overwhelming majority of peppered moths in northern England were light. As the Industrial Revolution swept through northern England, however, light moth populations dwindled as black moth populations grew. Overall, the total peppered moth numbers remained steady. So, before we move on, I am going to talk a little bit about the Industrial Revolution. You do have to know a little bit about it. So, in the 1850s, we were a largely agricultural economy, meaning most people lived on farms and farming was their main occupation. And then we started growing our or we started industrializing. So, we started building factories, and then people moved to the cities to work into the factories. Cities became super overcrowded, but also in the factories, we were burning fossil fuels, and fossil fuels are fuels such as coal and oil and these very old elements deep within our earth. And so we burn them, releases tons of carbon into the air, pollutes our air, and this is actually what has caused global warming. But for the sake of this question, you need to know that it talks about the Industrial Revolution. So if you look here at this chart, we have the year 1850. We have 10% black moths, which is pretty low. Light moths at 90%. In the 1900s, which is like when industrialization was really starting to take root, there was a lot of pollution at this time. Black moths were at 90%, and these light moths were at 10. So there's this reversal here. And what does that tell you? That probably tells you that these black moths seem to thrive in this dirty air. They do really well. Light moths don't seem to survive it very well. They need that clean air in order to survive. 1996, we had cleaned up our air. We passed the clean air legislation. It cleaned everything up. And suddenly, these guys, again, not doing so great. Fewer than 10. These guys, though, even better than before. In this same time period, the less industrialized rural England saw no such changes in moth populations. The only predators observed to prey on the moths were local birds. No migratory population changes of birds occurred during that time. British naturalist H.B.D. Kettlewell captured, marked, and released known numbers of black and light pepper moths in an unpolluted woodland and two similar groups in a polluted woodland. He later recaptured as many moths as possible. The following are some of Kettlewell's mark and recapture data. So we have Dorset, England here, unpolluted. So we have clean air. What can you expect? You're probably going to expect these light moths to be doing pretty well, and they do. He captured 496, or he marked and released 496, was able to recapture 62, which is 12% of those. But with the black moths, 473, only caught 30, only 6% survival rate. But here, we have this black soot and woodland, so very polluted air, heavy industrialization. And look at that, he recaptured 27% of these black moths, only 13% of these light moths. Again, these guys thrive in that awful air. What 20th century event most likely had the greatest impact on the recovery of white moth populations? Higher taxes imposed on oil imports, invention of the silicon solar cell in 1941, clean air legislation passed in the 1950s, or reduced fuel efficiency in new automobiles? So take a second to answer that. And the answer is actually C. It's that clean air legislation. If you were confused at all, read through these really carefully. The problem with D is this is reduced fuel efficiency. That means the fuel efficiency got worse in new automobiles and was polluting the air, which means that the white moth population would not have been great, right? Invention of silicon solar cell, no impact whatsoever. Higher taxes imposed on oil imports. Taxes have nothing to do with anything when it comes to this question. So the answer is that clean air legislation passed in the 1950s. Kettlewell's results indicated that black moths in industrialized areas had greater genetic variation than light moths, could outcompete light moths for food and habitat, had a higher survival rate than light moths in the same area, could change their phenotype when environmental conditions change. So this is the answer to this question is really easy. And this is one of those questions, and the OGTs do this a lot, where they want you to overthink it. Do we have any information about the genetic variation of light moths? All we know is that the black moth had one single dominant gene, which doesn't even really say they had, greater genetic variation. So we don't have enough information to know if that's true or not at all. We also don't know anything about the food or habitat of these black moths in industrialized areas. We just don't have enough information for that to be true. And then we're going to talk about phenotype here coming very shortly, the next slide actually. And I'll explain to you what that is, but that is not a possibility. It's actually literally just right in front of your face, as easy as it could be. Had a higher survival rate than light moths in the same area. That's it. They had a higher survival rate. When you see these questions and you see an answer that's very simplistic like that, ask yourself, is that a true statement? If the answer is yes, that's going to be the answer. These are making you think too hard. Okay, so that's another trick they do. The change in population numbers of black and light moths from 1850 and 1900 is most likely the result of the increased use of fossil fuels, solar energy, geothermal energy, or pollution control technologies. So if you were paying attention, I gave you the answer of fossil fuels already. That was the industrialization. We burned these fossil fuels and put those pollutants in the air. But I want you to notice something they did here. You have the right answer and you have the opposite answer right here. This is pollution control technologies is the opposite of the burning of fossil fuels, right? These pollution control technologies mitigated this. And then these two don't actually make any sense for the sake of the question. So they usually have a couple of fossil fuel questions on the test, and I'll go into that a little bit in a later slide so that you can always get those right. So we're going to switch for a minute. We're going to talk about Punnett squares. And I'm going to take the fear out of them for you. They're actually pretty easy, especially the ones that they use on the OGTs. But first, we're going to talk about the difference between a genotype and a phenotype. This is really important for you to know. There is a question about this on the OGT test. So a genotype, your keyword to remembering what that is right here, this gen, which is gene. You cannot see your genotype. It is embedded here in your DNA. So you can see, the only way you can see once the genotype is expressed, it becomes your phenotype. Your phenotype is literally what you can observe. It's what color hair you have, what color eyes you have, how tall you are. It's also the expression of certain genes that are in your DNA. That's why they use the sickle cell disease, right? That your genotype is located here in your DNA that's going to give you this sickle cell, but it's only your phenotype once we can observe it. So write that down. Genotype equals gene, phenotype, what you can observe. So we've got this Punnett square here, and the OGTs always use very simple Punnett squares. These can get super complex. The OGTs aren't going to do that to you. They're going to keep it simple. So we've got a blue-eyed parent here, and then we have a brown-eyed parent down here. So these two parents have a child, and this is going to be their genetic, the possibilities of the genetic variation. The way you put together a Punnett square is you bring this one down, and then this one over, and this one over, this one down, over, down, over, down. Now, the only way I should have explained to you that this is a recessive allele. So whenever you see these lowercase letters, these are recessive genes. Okay, so we have two recessive genes here. We have two dominant genes here. Well, anytime you have a dominant gene, it's always going to dominate. Meaning, if you have a combination with a dominant gene, that's what's going to win. It will always win. So if these two parents were to have a child, there would be a zero possibility for them to have a child with blue eyes because this parent here had two dominant genes. Does that mean that a blue-eyed parent and a brown-eyed parent can never have a blue-eyed child? Absolutely not. And I'm going to show you a really sloppy diagram of why that's not true. I'm going to do a Punnett square right over here. And then I'm going to have this blue-eyed parent up here. So that's two recessive genes. And then down here, I'm going to have a brown-eyed parent, and which they obviously have one dominant gene, right? Because you have to have a dominant to have the brown eyes. But they also carry this one recessive blue-eyed gene. So they carry this blue-eyed gene, but it's not expressed, right? Because what happens when you have one dominant gene? The dominant gene always wins. So let's put this together. We've got a dominant gene here, recessive blue here, dominant gene here, recessive blue here, and then check this out. We have a recessive here, and then guess what? We have another recessive here, and then we're going to bring this over here. So we have a recessive here, then we're going to bring this down and a recessive here. So if these two parents were to have a child, that child has a 50% chance of having a blue-eyed baby, right? So a 2 in 4 chance of having blue eyes. But these dominant genes, if they have that, it will always be brown no matter what. Okay, so I'll show you another example of that. Let's clear this and then move on. So we have some peppers here, and to be a green pepper, you have to have two recessive alleles. To be a yellow pepper, you just have to have one dominant. So this is really similar to the Punnett square I just showed you, right? So we have one recessive gene here. So if these two peppers were to reproduce, they'd have a 50% chance of being yellow and a 50% chance of being green. If you could see how we can make those combinations here. Now, if this were a dominant yellow, then they would have a 0% chance of having a green pepper baby. And since we're talking about chromosomes, I'm going to slip this little slide in here, and then I'm going to give you some keywords to always help you know if you are getting a genetic drift question. So genetic drift is basically, let's draw a diagram here so you can visually see what I mean by genetic drift. You have this huge mainland population here, okay? Let's just say this is, there are millions and millions of people here, and they have a dominant blood type of A. This is dominant. Does it mean that it's the only blood type? No, it just means it's the dominant. So most people have this blood type A. Well, a little small band of people move over here to this tiny little island here. And then over time, their blood type becomes B. The dominant type becomes B. What happens is these guys move over here. They're a very small subgroup of people. They reproduce only with each other. We're not introducing a lot of genetic diversity, and over time they they develop this different dominant blood type. So these rabbits here kind of show you another visual of how that happens. So you've got, here's your first generation here. You've got two white rabbits. You can see the different alleles, right? You have a dominant here and recessive here. And it's kind of all over the place. Well, all these guys have babies. And then let's look at this. We have more dominant genes here again. And we're getting less of these recessive alleles. So these guys reproduce again. And check that out. There's no recessive alleles. They were completely reproduced out of existence. So this is how evolution occurs. This is how new species occur. This is evolution. Now, how do you know if it's a genetic drift question? And when you go over the practice tests, and hopefully I'll make some videos about that, if you see the keywords isolated or small population, you know that question's about genetic drift. So make sure you write that down. That's going to be your keyword. So you can read a question and you will read all these things and you're like, I have no idea. But ask yourself, are they talking about a small isolated population where they're just reproducing with each other? If so, that's going to be a genetic drift question. So that's your keywords. Look for those, write that down because you're probably going to see that. Okay, let's get rid of this and move on. So we're going to switch again and we're going to talk a little bit about astrophysics. When they ask you about astrophysics, they're going to ask you usually about the same things. What we're looking at right here, this is called a spectrograph. Everything we know about heavenly bodies comes from these spectrographs. We study the light that these objects emit. We could tell a lot of things about an astral body off of the photons or the light that we receive. So they use these spectrographs. This is light, all of it. Some of it's light you can't see, right? But this is just the full band of it so that we can determine certain things. So we can tell how much of a chemical element is within a star because of this, right? Because different elements emit different light. And so we're able to figure out what elements are within that star or that planet and how much because of these spectrographs. We also can tell if that astral body is moving away from us or moving towards us. If it's moving away from us, it's going to be over here. This is called red shifted. You're going to run and write this down. If a star is redshifted, that means it's moving away from us. It has long red waves. This is how we know that our universe is expanding. There used to be two theories. One was that there's going to be enough mass in our universe that it's all going to collapse one day and implode, or there was not enough mass in our universe and it's going to continually expand forever. And so what they've determined is they found these spectrographs of these stars where actually these astral bodies are moving. And not only are they moving away from us, they're accelerating all the time. Our universe is getting bigger and bigger and bigger. This is how we know as we see this red shift here. Now, if something's coming towards us, it's a blue shift. And the one example I can give you of something that is coming towards our galaxy is there is a galaxy nearby. So it's called the Magellan Cloud. And it's actually there's a theory that it's going to merge in with our Milky Way galaxy. How do we know this? Because it has a blue shift. It's traveling towards us. Our gravity is pulling the Magellan Cloud into our galaxy, and they're going to merge. So that is the blue shift. When you see this question on the OGT, there's one about how can you tell such and such about a star? It's going to be the spectrograph or the spectrum. It's how we know everything is we study the light. And then this is just an example of how they figure out, and I can't explain it to you because I'm not an astrophysic, but how much copper is in a star, how much phosphorus and zinc and barium. And it's these dark spaces that tell them exactly what they need to know. So it's actually pretty interesting. It's how we can tell um, one other thing. One other thing about this spectrograph before we move on, we also can tell the age of a star because of this spectrograph, right? So if you have a blue star, those stars are young and hot, and they're going to die really soon. They're smaller, so they don't have as much energy to burn, and they die so much faster. Our star is about right here. Okay, so and our star is going to last about 10 billion years. We're about halfway through that cycle. As our star starts to lose its hydrogen content in its core, it's going to get bigger and bigger and bigger. It's going to turn into a red giant and explode. But this is how we can also tell the age of a star is the color. So we use spectrographs for everything. Now we're going to jump again and we're going to talk about fossil fuels for 1 second. I just want to tell you what they are, and I want to tell you how they impact our atmosphere and our earth. So they are natural fuels. They take millions, if not billions of years to form inside of the earth, right? So oil is actually formed like from lichen, which is a moss, and it takes hundreds of thousands of millions of years for that to turn into oil. So once that resource is gone, it is gone. We're not getting that back for a long time. It takes so long for that to form. Same thing with coal. It's they're fossil fuels. They're fossils, right? But when we burn these, we release all that carbon from burning them into our atmosphere, and this warms up our planet, and this is global warming. So we are really trying to get a control of this. And so when you see coal or gas or oil, this is a fossil fuel question. It's usually about temperature change, or it's usually about the damage that it does to our earth. Like we get acid rain from this that poisons our lakes. There's a lot of stuff that happens. So there's usually a few fossil fuel questions. Just know what they are and understand what they do. We're going to jump again. We're going to talk about energy. There's always a kinetic energy. Actually, there's usually this potential energy question on the OGTs, and the potential is exactly what it sounds like. These are actually pretty basic concepts. All you need to know is kinetic is literally just the action of moving, it's movement. Your kinetic energy is you walking down the sidewalk, right? Now, your potential kinetic energy is you standing at the top of a hill getting ready to roll down. Your potential kinetic energy is how fast you're going to go down that hill. So your potential energy is how much of an incline are you on? How much do you weigh? The more you weigh, the faster you're going to go. And here's a good visual of how to think about that. I've got this, I don't know what this is, a big metal block, and it's strung up by this wire here. The potential energy of this wire is how fast is this going to go down and hit this once that wire is cut. Potential energy, zero movement. But there's like a question, there's a skier at the top of a hill, and the question is, where is this skier's potential energy the greatest? And it's right there at the top of the hill before that skier is going anywhere. As he's moving up, his potential energy grows, but when he's at the very top, it's at its highest. And here's your kinetic energy, just literally the action of movement, the action of falling That's your kinetic energy. We're going to jump again. We're going to do a little bit of chemistry here. Got the periodic table. They don't usually ask too many questions about this, but I will tell you the things you need to know. Over here are the noble gases. And what's really important here, we've got helium, neon, argon, krypton, xenon, and radon. What you need to know about these is that these over here are what you will call stable elements. And I'm going to go into what that means here in a second. But they do not form compounds because their outer shell of electrons is stable. So you need to know that. You also need to know about this, but I'm going to go into depth about this. You need to know how to read this and what all these symbols mean. It's pretty easy, and I'll show you how to do that. Let me think if there's anything else I want you to know. There are some questions here. Sometimes I've seen it where you have Roman numerals here on top of these columns. If you ever see that, that represents the number of valence electrons in the element. I'm going to write this down for you so you can see, give you a visual of what I'm talking about. Okay. Now, a valence electron is the electrons on the outer, located on the outer shell of an atom. That's all that is. So these over here will sometimes ask about, they'll sometimes ask about the Roman numeral up here. This chart here doesn't have that, but if you ever see that, that's all that means. So let's move on. Now, I'm going to teach you how to read this. This is really, there's only one thing that you really, really need to know about this, and that's this number right here. And take your pen out and write this down. This is the number of protons, always. Okay, so you see this, and you'll see this question, I promise. What does this number represent? The number of protons in an element. It also is the number of electrons, but electrons get a little tricky and a little more complicated, and I'm about to show you why. This is the symbol name, C for carbon. Carbon is a super important element. It's what all living life is made of. It contains carbon. So they'll use carbon dating for testing, figuring out how old fossils are and things like that. So I've seen that question a few times. Now we're going to talk about this atom here so I can explain a little bit more about electrons. So how I like to think about this is a diagram of an atom. You have protons here, neutrons here, and then the electrons are on the outside. How I like to think of these is this is the mom and this is the dad, and they stay right here in the center. They don't ever move. Now a proton has I'm going to draw this for you. A proton has a positive charge, okay? And I'll tell you how important that is in a second. But a proton has a positive charge, but it just sits here with the neutron, which has a zero charge. These guys kind of control these electrons that I like to think of as little kids. So remember how I told you that prefix? Here's another one, nucleus, right? The control center. And in this case, this control center is the proton and the neutron. So these electrons are a little crazy. They do a lot of crazy things. If you ever see questions, and write this down, if you ever see questions about conductivity, or it is always an electron question. Electrons do all of the conductivity. They're the ones moving everywhere. These guys stay right where they are. They just hold everything together. Now we're going to briefly talk about ionic bonds and covalent bonds. I have seen this just a couple of times on there. What you need to know is a covalent bond shares electrons and an ionic bond steals them. And I like to think of these as relationships. A covalent bond is a very stable, nice relationship with two people living together and getting along, and everything's nice and stable. These ionic bonds, though, not so much. They steal from each other, and they're just magnetically attracted to each other. And I'll show you a little bit about that. But first, we're going to go over a covalent bond. This is oxygen. Oxygens are famous for their covalent bonds. And here's how this works. On your inside shell here, so we've got 8 protons, positive charge, 8 neutrons. On this first shell, you have 2 electrons. This first shell can only ever carry 2 electrons, right? So when we look at the helium, if you remember, has 2 protons here, which means it has 2 electrons. It only has 1 shell, right? Because it's got a proton or an electron here and an electron here. That's it. So it has 1 shell. Now this outer shell here, the second outer shell, always must have 8 electrons in order for it to be stable. Has to have 8, otherwise it's very unstable. And these guys always they'll do anything they need to do to be stable. Literally anything. And I'll show you what I mean. Now these 2 oxygen molecules here share a covalent bond. You can see 1, 2, 3, 4, 5, 6, 7, 8. They're just sharing these. This guy's sharing his and this guy's sharing his. They live very nicely together. They just get along really well. They're stable now because they have 8 electrons in their outer shell, so they're feeling really good about life. Now, they're also known as shared or bonded. Now we're going to talk about this toxic relationship, okay? Here we have sodium and chlorine. And chlorine is missing an electron. It's unstable because it needs that electron on this outer shell to be stable. Sodium here has 1 extra one. It's also unstable because its outer shell is not filled. So what happens? Chlorine steals that electron, makes it his, and then this sodium actually loses its outer shell. Well, what happens? Remember how I said protons have positive charges? Well, sodium now has 1 extra proton in there, which gives this molecule a positive charge because it lost 1 of its electrons. And this chlorine molecule now has a negative charge because it stole this guy, and now it's negative. So we got positive here. We've got 11 protons, 10 electrons. And then here we have 17 protons and 18 electrons. So this one's positive, this one's negative, and that is how they are attracted to each other. They're not joined together. Notice all that space in between them. That's why I call it toxic, okay? They have all that space between them. They're not sharing their space. They're just magnetically attracted to each other. Things to remember: protons and neutrons make up the center of the atom. That's mom and dad. Protons have a positive charge. Neutrons have no charge. Electrons have a negative charge. These are the crazy guys, and they are the ones doing all the movement. So you see those questions. Remember, that's an electron question if they're moving. Now, we're almost done here with the slide. I've seen a lot of questions about classification systems. I'm going to break this down for you and show you how to read them. This is kind of fuzzy or blurry, I'm sorry. But all living things are classified. This is how, and this has grown over time as we've discovered more and more organisms. And so we've had to create this huge classification system. So it starts with domain, which is eukarya. Does that look familiar to you? Remember those eukaryote cells from the beginning? Animal cells, right? Living cells. And then the kingdom, phylum, class, order, family, genus, species. Species is the most specific. Actually, you can go down to subspecies down here. But I'll show you I have an example of exactly what all this means. So we're going to talk about us, human beings, Homo sapiens. We're in the kingdom of animals. We are chordates because we have animals with a backbone. So that's our phylum. Our class is mammals. Our order, we're primates, but we're hominids. That's our family. And then our genus is Homo, and our species is sapiens. So we are Homo sapiens. So when you see these questions, that's what you're looking at. And I'll give you an example of what I mean. A student is studying several species that belong to the plant kingdom. Which 2 are most closely related? Ficus benjamina and Ficus lyrata, Castilla elastica and Ficus elastica, Bromos japonicus and Ipomea violacea, Formaldia pendorata and Ficus pendorata. Now, you can tell which ones are the most closely related because these guys here are in the same, I'm going to say family. I'm not actually sure. I'm pretty sure they're the same family. And then this is like the subfamily here. So, but the answer is A. But even if I didn't know that, which I actually don't know if they're talking about, this is a plant, obviously, but I could tell that this is the answer for a couple of reasons, even if I didn't know anything that I knew, which isn't much. This answer is crazy. So they always have a throwaway answer. This is your throwaway. Throw it in the garbage, okay? And when I go to the practice test, I'll show you what I mean. They always have these garbage answers. Now, here's another trick when you look at the OGTs that you know you can eliminate answers. These both cannot be the right answer, right? So you have these same subfamilies, Elastica, Elastica, and Pandorata, Pandorata. Well, these cannot both be right. They're both saying exactly the same thing. So A is the only answer that makes sense. So I hope that makes a little bit of sense. And then they're going to give you a chart like this sometimes. This classification system shows genetic relationships between 5 species of rabbits. So this looks complicated, this looks scary, but it's super not, okay? This is your family. It's the Leporidae family. This is the genus. It's Lepus here and Silvilagus here. This is the genus. And then we move into our species, right? So we have Calotus and Insularis, and then off of that Silvagus genus, we have these species here. It looks like we have a subspecies, and here. Which species likely harbors the virus infecting Silvilagus bachmani? So where's Silvilagus bachmani? Right here and right here. So which species, like there's a whole story that goes with this, but I just wanted to show you an example of how these look and how you read them. Is it Lepus calotis right up here? Probably not. Lepus insularis? Probably not. This is up here. This is a whole different genus, right? Silvilagus aquatics? Well, this is down here. At least it's in the same genus, right? Same species. Or is it Silvagus autobani? It actually is, right? This is the most closely related. It's literally the only answer. So if you take a minute to break these down, these aren't scary. Just pay attention up here, how they have them broken down, and you'll get those right. They're usually pretty straightforward, but remember, they're always going to try to confuse you. Anyway, that's the slide that I have. That's all I've got. It's really important that you also review the recordings I'm going to do for the practice test because I'm going to show you how to think through the questions so many times. The answers are embedded in the questions and you just have to know how to find them. But this should give you a good foundation and a good way to get started. So I hope you watch the other ones and then always reach out to me if you have any questions or you want some one-on-one tutoring. I'm always available. Thanks.